On behalf of the Biodiversity Institute, welcome to Lawrence and welcome to KU. We hope you have a very fruitful and engaging conference. Just a couple of quick thank yous. Firstly, thank you to Jayhawk Hospitality, who were amazing in helping arrange all of the details of this conference. And also to our volunteer crew, who you will see helping out in all of the rooms here and also at the museum this evening at the Icebreaker. So a couple of local logistics. The important things first, bathrooms. Bathrooms are right outside the door here, just to the left, and there's another set down the other side. Wi-Fi, you can get onto the KU guest Wi-Fi. There should just be a place where you can say, I accept, and then you can get on, or you can use EduRoam if you have that capability. Lunches. So we have 2 food trucks that are going to be coming out here for lunches. We're in a bit of a food desert in the middle of KU between semesters. And so we'll have 2 food trucks that will be coming out. Bryce's barbecue food truck will be here today in the parking lot just on the other side of this building over here. They've guaranteed us that they will be able to serve everybody in an hour and a half. So hopefully they can get through everybody. There are vegetarian options, even though it's a barbecue food truck. Tomorrow we have M&D's Mexican food truck, and there's a couple of options there, including a vegetarian option as well. For those who are unable to find anything at the food truck to eat, there is also Miss E's, which is the sort of local place where you can get food up at one of the dorms up on the top of the hill. If you need directions, either chat to me or chat to Jill, and we can direct you up there so that you can find something else for lunch if you can't find anything at the food truck. We have to meet a minimum with them, so we would prefer for people to eat at the food truck if at all possible. Transportation. Apologies for the buses this morning. Apparently we got very late notice that the bus could not make it into the parking lot. So there was a bus stop just outside the parking lot. So tomorrow morning, go to that bus stop just outside the parking lot and you will be able to get to the venue from there. There will be buses departing from here at 4:30 to take people back to the hotels so that you can chill out for a while before you come to the Icebreaker. And then the buses will pick you back up from there and bring you up to the Icebreaker at 6:10 for a 6:30 start at the Icebreaker, which is just outside of the museum. Tomorrow morning there will be pickup again at 8:10 outside of the hotels to bring you back down here, and then there will be departing buses at 4:30 and 5:30 because we have 2 sessions ending at different times that will take you back to the hotels. And the buses will sort of congregate about where the food truck is and pick you up from there. Friday, there are no buses available. There's just the IDIC Bio workshop and various other things. But if anybody needs a ride to the dorms or to the hotels, please let us know and we can arrange something. The Icebreaker reception is at 6:30 on Asher Plaza, which is just outside the museum, as I mentioned. We'll have heavy appetizers, beer that's been donated by Free State Brewery, wine and soft drinks, and hopefully everybody will have a good time. The museum will be open for the duration of the Icebreaker, so feel free to grab your favorite beverage and take a walk through the galleries in the museum. We will also have collection tours at 7 and 7:45 for the collections that are in Dyke Hall. Those that are not in Dyke Hall will have tables in the panorama exhibit exhibiting some of the specimens from their collections. So we hope to see you there. It should be a lot of fun. And then the buses will depart from there at 8:40 to take you back to the dorms and back to the hotels from there. If you have any problems with dorms, with the hotels, with transportation or anything, please let one of us know and we can try and sort it out for you. So with that, I'm going to hand over to our outgoing director, or Jorge Soberon, to say a few words.
Good morning, and first of all, thank you to all of you for being here. This is a great turnout, and I'm very glad to see all of you here. I am still the director for another 4 weeks, and then Nico, Franz will become the director, but more about Nico in a second. First, I would like to tell you about how excited we are about this conference. KU has always been interested in the combination of computer technology and ideas with collections. Since the days of numerical taxonomy, which probably many of you don't even remember, but we were very much in the thick of it in the 60s and early 70s of last century. Collections and IT are born to be together. The amount of value that is added to collections by adding enabling them with the computer technology is just amazing. In the case I know best, which is the Mexican case in Conavio, the fact that the collections were computerized and digitized and made available through the internet made them immediately available to a huge amount of students in universities, NGOs, small government agencies. It was just astonishing to see how much use was made of collections, which in the mind of most people, and including many scientists, the image that invoke is dusty bookshelves or dusty shelves with old Victorian scientists doing something which is boring and uninteresting. Well, collections are absolutely essential to work in science, in biology today, and with the extra tool of bioinformatics, they become amazingly powerful. So thanks for doing this for all of us and for all the users that some of you may not even realize that you are enabling some youngster in a high school which is downloading data and doing things with that data. The next director of the Biodiversity Institute is Dr. Nico Franz, who's also very much into the thick of it. He has been not only interested in implementing ideas about biodiversity informatics, but also in developing the concepts, the ideas of that. He is, from my perspective, doubly interesting because he's an entomologist, and I am an entomologist. And not only an entomologist, but an expert on curculionid weevils, which are amazingly varied and numerous and interesting and important from an economic perspective. So, well, without further ado, I would like to give the floor to Nico, and please join me in.
Right, I get 2 minutes here. Hi, digital data. Hi, greater IDIC Bio community. Thank you, Jill, Andy, and Jorge. Very gracious. I suppose I'm 2 for 2 as a person welcoming you in some form to digital data, as I'm moving this summer from my position at Arizona State University to become the new director here at the KU Biodiversity Institute and Natural History Museum. Digital data is among IDIC Bio's most valued traditions. We're thankful to IDIC Bio and to the Biodiversity Institute for your continued support and excellent organization. We can all look forward to a strong and diverse program of presentations and activities in the coming days. And I hope that for those of you who can be here in person, you also find the time to experience Lawrence and KU's exceptional commitment to science, education, and societal impact, broadly speaking, but most definitely in relation to systematics, collections, and biodiversity informatics. So much that our community, and Jorge alluded to this, cares about was started here, including Specify, and continues to be advanced in new directions. So I'm proud and thankful to soon be part of this community and trajectory. Thank you and welcome again.
Alrighty, so I'm your moderator too for this session. So let's get this party started. So a reminder to the virtual audience, if you have any questions for the presenters, please put them in the Q&A. We have somebody here who will be monitoring the Q&A and will hopefully give your questions to the presenter at the end if we have time. A reminder to all of the speakers in this session, you have 10 minutes. I'm going to stand up at 8 minutes and stand at the end of the podium just to make you feel bad. And then I'm going to cut you off at 10 minutes. And we will have 5 minutes after that for questions. We're going to try and keep things on track so that we can keep all of the concurrent sessions going at the same time. A reminder to all of the panelists to please mute your mics and stop your videos if you're not presenting so that we can see the presenters on screen. So Our first talk this morning is from Town Peterson, and he's going to be talking about blockchain-based prototype solutions for biodiversity data management. Town. There's a clicker if you want to use it, otherwise you can just use that for us. I'll give you a couple more.
So, welcome everybody. So I'm sitting over there and I hear Andy say, you've got 10 minutes and a couple for questions, right? How did that feel? Yeah, I know. Anyhow, it's one of those nightmare scenarios where you start off like with this cold feeling, right? Welcome to Lawrence. Those of you who don't live here, welcome. I hope you enjoy our community. It's a place we're all very proud of and very much enjoy. And then I'll go from that to an apology, because usually when I give a talk, I try to talk about something fun in biology. And I was asked to give a talk in this meeting, and I laid out some topics that might be interesting. And what gets chosen is blockchain, which is fascinating. I've had a lot of fun with this over the last few years, but there's nothing to show. So I'm going to apologize to you all that this is all words and it's not images. And I'm going to apologize that there won't be any like biology message. So I'm just going to trust that you all are tanked up enough on coffee that there's not a problem and you'll make it through this. And then we'll get on to some other talks. Yep.
There we go.
Okay, so now you can see my presentation. Thanks, Andy. Essentially, what I'm going to talk with you about today is a sequence of work that has been developed because a major player in the blockchain world happens to be a KU engineering graduate. And so there is a group that puts out essentially block grants and funds interesting work. I hope this ends up being interesting, but work that goes outside of just using blockchain for for banking transactions and things like that, and asks what more could this sort of work be useful for. So you'll see this is a collaboration between a couple people in our Department of Electrical Engineering and Computer Science, especially Professor Drew Davidson, and then also in the Biodiversity Institute, myself and Fernando Machado Strevel and Ali Khalifar, both recent PhDs and departed from here, but both still involved in the project. So if you come over and join us tonight at the museum, I'll be excited to show you some neat bird specimens. They're all neat, the bird specimens, that is. But we're focusing on those little white pieces of paper that are tied to each one of them. And they have a very, as you all know as well as anybody, they have a very complex set of information associated with them. And I just took these pictures just to give you an idea of this. Bird specimens are different from mammal specimens. Mammal specimens are different from plant specimens. They're all different. But we have information about the taxon. We have a catalog number. We have the people involved. We have body mass and body condition, information on sex and reproductive condition. So we have this ton of information, and a lot of it needs to be interpreted, and a lot of it can be reinterpreted through time. So notice this was originally identified as Pipilo aberti, and then somebody along the way, probably this HWL person, came along and re-identified it as Pipilofuscus mesoleucus. Okay? This is what we do in museums. This is the Victorian scientists puttering around in the collections that Jorge mentioned. And for about, well, at KU, the last 40 years, because the bird collection was computerized as of about 1981, but we've been capturing these data digitally, and that involves a bunch of steps that you all are familiar with that end up producing some sort of table or database. In this case, it's a view of Specify. So that's kind of things that you all know, but there are some problems. There are some design flaws in how we're managing biodiversity information at the moment. So I just showed you a little bit of an example of how biodiversity information records are dynamic. The content evolves very quickly when a specimen comes into the museum, and then it kind of evolves in lurches and starts and stops from then on. It might be a re-identification, georeferencing, all sorts of things. Here's a big one. In our current data management world for biodiversity information, the data exists as a single final version. And that's a big point. I know in Darwin Core there's the verbatim field, but that just says that there might be 2 points. And yet data evolve as a continuous process. And so I don't think even 2 points are enough. And perhaps more seriously is we don't keep a record of what that previous state was. And so I might re-identify a specimen, and then 10 days later or 10 years later, somebody else might re-identify it again. And we've lost that sequence of steps, yet there is information in there. And then on a more individual basis, we don't have tracking of who did what. So it's hard to say, oh, individual 1 georeferenced these 50,000 records, individual 2 updated taxonomy on these records. I know there are solutions in some platforms to this, but what I'm looking for is a full solution. So how do these data records evolve? You know this. One important concept is the extended specimen. This is a pretty famous image that Dave Blackburn and colleagues developed to justify the OVERT project. And little by little, this one frog in the middle of this has had data added to its digital existence in many different realms. It might be a genome sequence, it might be what have you. But that evolutionary process of the data is pretty important. The taxonomic changes, this is one that I literally, even though I'm a professional ornithologist, every time I need to refer to one of these siscins, I need to look up what is the genus of the week for this group. They're just things that aren't stable. So we started looking at this idea of blockchain technology. And I'm going to go through what is blockchain because I didn't know well what it was when we started, and I'm still at a low level in terms of the actual IT dimensions. But a blockchain is a distributed ledger, so distributed. It has a list of records, which are the blocks, and the blocks are linked together by codes that essentially tie this one to its antecedents and to its successors. Each block has information about the preceding block, and so that's the chain part of it. And so the neat thing is that these unitary blocks, which are in essence transactions about the data for a particular specimen, these unitary blocks allow us the possibility of going through and querying across the whole history of data management for a specimen rather than just the final version. So then the question is, what could we get out of this for biodiversity information management? The promise would be full tracking of all changes to a data record through time, keeping all of the versions of all of the data fields with full attribution as to who did what. We would be able to realize that we made an error and go back 3 steps to before a particular change was made. We could attribute credit, and it doesn't matter where the person is or who the person is. We can make full accounts of who did the georeferencing for this data set or who did the taxonomic updates through time. And we can also have different user types and levels with different levels of permission. You see, I told you needed that coffee. It's right over there if you need some. Okay, this is early in the morning for a talk like this. So essentially, we've set out to illustrate this technology. The people at EECS developed a prototype. It's a pretty basic prototype. It's not something that you show off yet or you distribute yet. It's just a prototype to explore this. So that's a platform. And then we decided, well, we need an example, essentially a case study. And we thought about a bunch of different examples. You know, the whole BI is too much. The whole bird collection is too much. The favorite phrase in the bird division is kill 2 birds with 1 stone. So we have a project that is called the Bunker Resurvey Project, where essentially we're setting out to revisit and recollect all of the sites across Kansas and eventually across the Great Plains that were collected and surveyed back 110 years ago by my ancestors, especially a guy named Charles Dean Bunker. And so we decided, let's take the Great Plains material from before 1950 and let's clean up and enrich all of those data as a team effort. So we created different roles. We had everything from database manager who could do anything down to student level, undergraduate student level, where any change they made had to be okayed by somebody at a higher level. We imported the data from a CSV file output from Specify. And these are the data that we put in, which were the holdings from across the Great Plains. KU has a good portion of this. The Great Plains are a mixture of open areas and wooded areas, mainly riparian. Those are the official EPA limits of the Great Plains region. And so we put in all of the Great Plains holdings of birds at KU, 23,000 records approximately. And then we checked with the graduate students around the 7th floor of Dyke Hall, and we said, who's going to be around for a couple of weeks over Christmas break and would like a fun time in the bird collection? And we actually assembled, I believe it was 22 people, because bird specimens are fun. And they spent the Christmas break, working through the specimens one by one. They ended up handling about 17,700 specimens over about a month. They also, a different group ended up working through georeferencing all of this material. The georeferencing for some of these sites is a pretty laborious process. This is a neat illustration. They're not exactly oriented the same, but here's the Missouri River and here's the Missouri River. But the Missouri River decided to change its course. It used to run like that. You can see Independence Creek here and here. And so figuring out where this site was not simple. Many of you know what I'm talking about. But all of the localities for KU birds from the Great Plains got worked through and georeferenced. And corrections were made to each record as either information was added or as information was corrected. And so this is just an example of one of the big tasks. You can see this is a very exciting collection of house sparrows. But what was very useful information that was added was information from the specimen about age. And this is information that wasn't consistently captured when the data were captured from specimen labels. This required a combination of plumage and label information. But it's really important because it establishes what we know about whether the species was breeding at that site. So it'll be really important to the resurvey project. So that's about as exciting as this talk gets. But we had 22 people participating in developing this prototype. There were all sorts of different roles, from people who could do anything to people who had to be supervised at all times in terms of the data. 17,677 specimens at KU were reviewed, and for each one of them, the species ID was checked, the sex was verified visually and via the label, which sometimes picks up on disagreements, and most importantly, the age information was captured. All of those specimens were georeferenced using the point radius method, and full metadata were added to the record. Names were standardized. That was seemingly small, but it does help to be able to go back and recheck things or summarize all of the specimens that we might have from a particular collector. Dates were preserved and unified in a variety of formats. Preparation types were standardized. So in essence, what we were able to create is a comprehensive data set ready for use on the science end, but also we have a full record of kind of the evolution of this data set. And so that's what we're looking to illustrate on the blockchain side. Where is this going? It's only a prototype. I'm definitely not going to show it to you today. It's not ready for use, but it is a useful example. We're certainly going to need quite a bit of development and testing to be able to optimize the design. Pretty soon we'll be looking to put in a proposal to the NSF Infrastructure Innovation Program. But essentially the thought is that if at the end of this long process of design and refinement, a blockchain style technology could go as a back end behind any or several of the front ends that have already been developed. So that's essentially where this effort is. If you have any questions, I'm happy to do my best to answer them. And sorry for a talk that didn't have much biology in it.
Thanks, Tom. Any questions?
Yeah, thank you for this. I think it was a very interesting talk, and yeah, I think the question of how we handle versioning in transaction records in biological data is really important. But I guess I'll ask the slightly unfun question, which is, because I'm a backend person, I am a database person working with Morphosaurus. Why blockchain compared to implementing a similar transaction record in a traditional database? I mean, I feel like we've looked at blockchain and it seems like it tends to be slower and more inefficient, and it has a larger carbon impact in terms of electricity usage. So from your perspective, like, what are you getting from using blockchain compared to implementing a similar system in a traditional database?
I think, first of all, I'm not a back-end person, so I can't give you a technical answer. But I do see a lot of value in exploring multiple solutions. And notice I didn't say blockchain. I said blockchain inspired or blockchain style. And so essentially what I see this as is a handshake with a community that is seeing a lot of use and development in other areas like finance. And what can we as a biodiversity community learn from that? Is it the final answer? No. Am I saying it's the best answer? No. I'm just saying it's worth exploring. Yeah. You're going to get your steps in today, Andy.
Hi, thanks, Town. I'm curious about your thoughts at the level at which you've applied this idea. So what if you are thinking about once your data leaves the building and gets out into the wild world, other people want to do the same thing that you were pointing out, right? They want to point out issues with the data, or they want to add their knowledge to that data and give you back that information. So have you thought about that level in your model?
Yeah, a really good question. And in fact, Andy and I and a couple other people put a lot of thought into that. Essentially, we could imagine doing this at a more meta level where the data that are contributed by individual institutions are pooled. Imagine what IDIG-Bio. And then all of the improvements and such that get made to those data are tracked using these blocks. Perfectly good idea. The one thing I always come back to is I want to make sure that the data improvements get back to the original institution. So I think both may be very interesting to explore, but to me as a museum curator, the more compelling one, at least to start with, was within the institution. Doesn't mean it's the more important one. Yeah.
We have one online, Town. Why use blockchain and not only a Merkle tree data structure? Blockchain has some security limitations that made it power consuming.
Again, blockchain inspired, blockchain style. I'm not competent to answer that question. I can certainly connect the person who's interested in that with my colleagues in the ECS, and they can do a better job of answering at that level.
Any other last questions? Okay, thank you, Tom.
I got a lot into 5 minutes, didn't I?
All righty. Kelly is on PowerPoint. That one. That one. Alrighty, our next speaker is Kelly Speer. who's going to be talking to us about hosts, parasites, and microbiomes, a system for studying natural complexity in a changing world.
Thank you so much. Okay. Essentially, a summary title for this talk can be Come into the Rabbit Hole with Me. Natural history collections are continually reinventing themselves. Collectors depositing specimens in the first natural history collections in Europe hundreds of years ago could not have imagined the way those same specimens are being used today. Through the advancement of next-generation sequencing technology and imaging technologies like micro-CT scanning, we're now able to derive a lot more information from these same specimens than we have ever been able to get before, and that were even imagined when these were collected. We're going through another kind of renaissance with this idea again through microbiomes and pathomes. This is a fluorescent in situ hybridization image of the midgut of a fly. All of the fluorescing red components are bacteria in the midgut of that fly. So we're now understanding that these individual specimens are much more than a representative of a single species. Take, for example, this bat voucher specimen. This bat voucher captures components of the DNA, RNA, and morphology of this bat. But it also captures endoparasites, ectoparasites, the microbiome and pathome, and environmental data. This collective unit has been termed the extended specimen, and we can dive a little bit deeper into these interactions through new advances in our ability to sequence and image these extended specimen data types, and also to database them. At the Michigan Pathogen Biorepository, where I'm currently working to develop this new type of collection, we're focusing on cataloging and databasing these pathome and microbiome components of the extended specimen. And we run into a lot of challenges that I'd love to get this community's feedback on in terms of databasing and what sample types we should collect, how we can make these more discoverable and interoperable. So why is the extended specimen important? Well, if we know that an individual specimen or an individual organism actually represents and captures a lot more than information about this single individual, it captures a whole community of microorganisms associated with it, then and these microbiome communities impact fundamental components of a host biology, for example, immune function, health, reproduction, even behavior, then we need to capture and understand this microbiome pathome extended specimen in order to tackle the big questions in biology, like what are the consequences of environmental change for biodiversity loss, what determines the susceptibility of a host to a pathogen, and how can we predict and prevent the next pandemic. In my own work, I focus on bats and their associated arthropod vectors and the microbiomes associated with both. This is where we're going into the rabbit hole. So I'm telling you to imagine a bat, and then imagine a vector on that bat, and then imagine microbiomes in that vector and with that bat that are all really important in reflecting the community of organisms in the space of environmental change. Some of you may be familiar with bats, but I want to take us through this system a little bit. So bats are ecosystem service providers. They are seed dispersers and pollinators and insect pest controllers. They're also models for immune function. They're able to host many pathogens without showing incidents of disease. For example, some pathogens that are almost always fatal for other mammals, bats are able to tolerate, for example, rabies virus. This makes them really interesting from not only this ecosystem service perspective, but also from this pathogen surveillance perspective. Bat flies, I've circled one on the head of this bat, are obligate blood-feeding ectoparasites of bats, and they vector pathogens to their host bat. For example, there's bat-specific malaria. Bat flies also vector viruses and bacteria. So that's where we get this vector component. And they are really closely related to some other interesting flies. So there are two families of bat flies, the Nycterobiidae and the Strepilidae, which I've highlighted using stars. They're closely related to the Hippobosidae, or the KED flies, which are vectors of agriculturally important pathogens like blue tongue virus, and also to the Glossinidae, the tsetse flies, which vector important human pathogens in Africa, like sleeping sickness. Just an additional note, I'm going to be using these two silhouettes for the rest of the talk to indicate these two families of bat flies. So by studying bat flies, we're actually able to make some conclusions or hypotheses, or derive some new hypotheses about these other closely related flies that may be relevant for agricultural transmission of pathogens and human pathogen transmission. Okay, so what about bacteria? Well, bats may rely on bacteria to provision nutrition that's missing from their diets. For example, some bats have really specialized diets. They pretty much only feed on sugar water, and it's been hypothesized that the microbiome can help provision nutrients missing from that specialized diet. But this interaction between bacteria and a host is much better described in bat flies. Like all obligate blood-feeding flies, bat flies have primary bacterial endosymbionts that provision B vitamins that are missing from the diet. Blood meal doesn't have a lot of B vitamins, so these bacteria are a requirement for survival and reproduction in bat flies and all other obligate blood feeders. By studying the interactions between bats, their vectors, and the microbiomes associated with both, we have a really convenient nested multi-trophic system. It's essentially a community of interacting organisms, and we can use this community model to examine how environmental change impacts communities as a whole. This is a really hard question to get at typically, but by taking advantage of this kind of host parasite microbiome system, it's nested, it's easily dictated, so it has some nice advantages. So the take-home message is essentially the extended specimen enables novel insights in ecology and evolution, while also presenting challenges in terms of how we capture these specimens and database them. I'm going to take you through three kind of vignettes that demonstrate this value of the extended specimen. One looks at how geographic barriers influence population connectivity and draws on some pretty seminal papers on host-parasite interactions. The second, except it says one again. The second is how does geographic distance influence biological communities? This is a map of the invasion and spread of the fungal pathogen white nose syndrome in bats across North America, starting in the northeastern United States. By taking this extended specimen perspective, we can better predict and potentially prevent the invasion of white nose syndrome into naive bat populations. And the last, again, labeled one, examines how does habitat loss influence communities of interacting species. We know from model systems like mosquitoes that the presence of specific bacteria can actually impact their vector competence. So when mosquitoes are infected with a specific type of Wolbachia bacteria, they are resistant to malaria infection and less competent vectors. So this extended specimen has the power to inform our understanding of pandemic potential. Okay, so let's start with our first example. How do geographic barriers influence population connectivity? For this little vignette, we're going to be talking about islands of the Bahamas. These islands are split into two banks, the Little Bahama Bank and the Great Bahama Bank. And these banks of islands are separated by a deep oceanic channel called the Providence Channel. This Providence Channel has prevented these banks of islands from ever being connected, even through glaciation cycles in the past. When we look at a really good disperser, Tadera brasiliensis, the Brazilian free-tailed bat, which is migratory in portions of its range, it can fly really long distances. We see that the Providence Channel, indicated by this red arrow, is a barrier in the mitochondrial DNA of these bats, causing differentiation between the populations on the Little Bahama Bank and the Great Bahama Bank. We confirm this using nuclear microsatellite data. The red arrow again shows the Providence Channel, and these two populations are almost completely distinct. And we also use museum specimens, morphology from museum specimens to confirm that Little Bahama Bank bats look different than big Great Bahama Bank bats. So this is pretty surprising because the Providence Channel is not very wide. And as I said, the Brazilian free-tailed bat is an excellent flyer. However, when we look at patterns in another bat in the same island system, the Buffy flower bat, which is an island endemic, we see a much different story. we see a lot of population connectivity across the provenance channel. So we wanted to examine this with a higher resolution marker. We decided to compare the population genetics of the buffy flower bat with its associated batfly ectoparasite. So when we look at the population connectivity of the buffy flower bat, dispersal is high across the provenance channel. We see 10 individuals per generation going across the provenance channel, which is what we expected. It gets interesting when we look at their ectoparasites. We see much lower dispersal across the provenance channel than in the host bat, only 1 individual per generation, which could indicate that there are a few things happening here. So bat flies may detect dispersal patterns that are difficult to assess in bat genetics alone. Bat flies may be reflecting a recent reduction in bat dispersal across the provenance channel, or bat dispersal could be rare, but many bat individuals are dispersed at a time. For example, hurricanes have been shown to be a really important mass dispersal event of bats across the islands of the Caribbean. But neither of these patterns would be apparent if we only had looked at the bat genetics. This indicates the importance of having database structures that can link the genetics of both the parasite and the bat to each other so that people can get the complete story about what's happening with populations of interest or conservation concern. This can be a database challenge, although some collection management software do a good job of connecting genetic data from host and parasite. Okay, let's move on to our second little vignette. How does geographic distance influence biological communities? For this vignette, we're going to be talking about the lesser long-nosed bat, Leptonicterus yerba-buena, and its face is totally covered in pollen because it is a pollen and nectar-feeding bat. It is the primary pollinator of cactus and agave in Mexico. So if you like tequila, you like this bat. The nice thing about this bat is that it's migratory. So it spends the fall and winter in central Mexico and the spring and summer in northern Mexico and the Baja Peninsula. Because it's such a good disperser, we can control for the impact of dispersal on differentiation in the microbiome communities across these sites. We can essentially isolate, is it the environment that's impacting the microbiome communities, or are the microbiome communities inherited or filtered or specific to a host? In other words, we can test the hypothesis, is the bat skin and bat fly microbiome, does it vary geographically, or are they conserved, indicating potential selection? So what we did here is we swabbed the cave indicated by this trapezoid and the bat skin, and then we sampled the microbiomes from 2 bat fly species associated with this bat, indicated by these 2 silhouettes. What we found is a lot of differentiation between these microbiomes. So let me get you oriented to this plot. This is a principal coordinates plot. So here each point represents a microbiome, and when 2 points are close together, it indicates similar microbiome communities, and when they're far apart, it means they're very dissimilar. I've colored these by their sample type, and what you'll notice is that the cave and bat skin microbiomes in this kind of yellow and salmon colors are very similar to each other, almost completely overlapping. But the microbiomes of the 2 batfly species associated with these bats and in the same caves are extremely different from the environmental microbiomes and different from each other. We can dive a little deeper into this using qualitative plots. So here, these are relative abundance plots. Each of the colors represents a different bacterial taxon and their relative abundance in these different sample types. And each bar represents a different sampling locality. So here, on the left, is this gonna work? Yes. On the left, we have the microbiome composition of the bat skin microbiome at northern Baja. And on top, we have the northern Baja cave microbiome. So this matches what we saw in our principal coordinates plot. Our bat skin and cave microbiomes vary a lot geographically, and they also are similar to each other at a sampling site. So northern Baja bat skin microbiomes look very similar to northern Baja cave microbiomes. When we compare this to the 2 batfly species, we see this, a different story. Not a lot of geographic variation, and very different microbiomes from the cave and bat skin microbiomes. And I want to point out one other interesting thing. There are 2 bacteria that have much higher relative abundance in the batfly species Trichobius sphaerinotus on top than they do in the second batfly species. Even though these bat flies are feeding on the exact same bat blood, they're in the same caves. These two bacteria are Bartonella in the solid line and Wolbachia in the dashed line. Bartonella is an arthropod vectored pathogen of mammals. Some members of this genus cause human disease. For example, Bartonella quintana causes trench fever. And bat flies are the primary vectors of Bartonella in bats. Wolbachia, on the other hand, I mentioned a little bit earlier, is a reproductive parasite and sometimes facultative symbiont. Some members provide protective capacity against malaria in mosquitoes. So it's really interesting that these two bacterial taxa are higher in relative abundance, even though so much of the environment and diet of these fly species is controlled and the same. So bat fly and bat skin microbiomes are distinct and variably reflect geography. Bat fly microbiomes are distinct from their environment and bat, and distinct from each other. So this indicates that the bat fly microbiomes may be strongly conserved, as we would expect, given they rely on a lot of these bacteria to provision nutrients missing from blood meal. On the other hand, there's strong geographic variation in bat skin and cave microbiomes despite high dispersal between these sites, indicating that potentially the cave microclimate is a really important determiner of what's happening with the bat skin microbiome. So we used swabs as a really valuable resource for extended specimen data in this sampling effort. We used swabs of the bat skin and cave, but these are not typically sample types that are collected by natural history collections, even though they provide valuable information. And in this case, they're acting as our voucher of the microbiome. So we have to get a little bit more flexible about how we're thinking about vouchers based on what we're trying to catalog. Okay, let's move on to the last story. How does habitat loss influence communities of interacting species? This is a map of forest loss through time overlaid with human population density. Higher intensity forest loss and higher intensity human population density are darker pink. This forest loss is expected to drive a destabilization of ecosystems that has been associated with pathogen spillovers from wildlife to humans or agricultural animals in the past. So studying this type of habitat loss that creates these opportunities for spillover is extremely valuable for that question of how do we prevent the next pandemic? So by looking at this bat fly microbiome system, we can examine how environmental change is impacting parasite-associated microbiomes, which can be relevant for vector competence. And we can use island biogeography theory as a null hypothesis for how we might expect these parasite-associated microbiomes to change in response to habitat loss. So according to island biogeography theory, smaller isolated habitat patches support fewer individuals and less diversity of species than intermediately sized habitat patches or the source patches, which support the most number of species and the most abundant individuals. Okay, so for this project, I really want to thank my collaborators, Thiago Teixeira and Alexis Brown. Thiago spent a year in the Atlantic forest sampling bats and their ectoparasites from these different sites. So the big swath of green at the top of your screen is actually a protected reserve called Regawa that he sampled 3 times. All of the green patches outside of that big swath are actually remnant forest fragments separated by agricultural land. And the nice thing about this system is that these little patches vary in their size and isolation from each other, lending themselves to that island biogeography kind of hypothesis. Okay, so we selected parasites from the most common 6 species of bats across all our sampling sites. These include fruit-eating bats, blood-feeding bat, and insect-feeding bats. And we got their microbiomes from their associated bat fly species, 222 microbiomes in total. Okay, so what did we find? Well, bacterial richness does not vary with habitat area or isolation. On the x-axis here are our sampling sites ordered from largest on the left to smallest on the right, and the y-axis shows bacterial taxon richness. If our parasite-associated microbiomes were following expectations of island biogeography theory, we would see a strong negative trend here, but we don't see any trend at all. However, this is only one component of variation of the microbiome. Another important component of variation is relative abundance. So in order to examine how relative abundance changes in response to habitat loss, I used a type of ecological interaction network called a bacterial association network. I know this looks like a bunch of spaghetti on the wall. Don't worry, we're going to go through it. So each of these dots here represents a bacterial taxon. And when 2 points are connected, it indicates that those 2 bacterial taxa have correlated changes in their relative abundance. Now, these correlations can be because of an interaction between these bacteria or because they're synchronously responding to a host or environmental cue. So what do our networks look like from Regua, our protected site? All of them have these kind of isolated neighborhoods of interacting bacteria called modules, which, network lingo. And they look very different from our networks from our small patches outside of Regua. This is just 2 examples out of 10, and they all look like this. Instead of isolated neighborhoods of interacting bacteria, we have these long band-like networks. And it's been hypothesized that these long band-like networks are more susceptible to environmental perturbance, because if you imagine severing the connection between any given bacteria in our top graphs, that only impacts the immediate module or a few bacteria in the network, so very little impact on the global network. However, if you sever a connection in the lower networks, that impacts the connection. It essentially severs the connection or the network in half. It has a much larger impact. So what this indicates to me is that there are cascading consequences of habitat loss in communities of macro and microorganisms. So parasite microbiomes variably reflect environmental change, and this impact of environmental change is driven by habitat patch size, dictating the community of bats that can occur in that habitat patch, and therefore the community of parasites that occur there, and subsequently the community of microorganisms. However, there could be a direct connection between habitat patch size and parasite-associated microbiomes through the impact of temperature. We know from ticks that as the temperature increases, the microbiome enters dysbiosis. So there could be this direct connection. Microbiomes may not follow null hypotheses established for animals and plants, so there's lots of room to develop new null hypotheses for microbiome communities. And ecological networks offer a future route for examining these nested communities in changing environments, but how do we database these types of communities and their putative interactions? These interactions can be really important in capturing change through time that isn't captured just by looking at bacterial taxon richness. Okay, so I hope I've convinced you that the extended specimen enables novel insights in ecology and evolution. If you want to learn more about how we're tackling the databasing problems that I brought up throughout my talk, I would encourage you to go to Summer Mangarelli's talk today at noon in room D. Thank you so much for your time, and I can take questions.
Thank you, Kelly.
Thanks for the awesome bat inception talk. That was cool. So, decades ago or whatever, we weren't even thinking about like collecting tissues, for example, but now it's like a critical part of collections infrastructure. Like, what do you think we need to do to have critical infrastructure for like collecting these types of data, like basic minimum?
Yeah, I mean, I think a lot of different collections are thinking about this, like how we need to take as many different sample types as possible. The challenge comes in storage space, honestly, where if you decide that you need to take oral and rectal swabs and skin swabs from everything, you're talking about 10 to 15 tubes for each voucher that you're taking. And at that point, it becomes an exponential growth issue. So I think a lot of people are thinking about this. I think what we need to become or develop our creative thinking skills is how we're going to store all these sample types.
Good morning. Great talk, Kelly. So, I may have missed this because I got in a little late, but did you look at the gut microbiome in association with the ones with the various parasites, and do they differ as association between, if you get bit by one, does it alter the gut microbiome or the, oral microbiome or something like that of the bat?
altering the gut microbiome of the bat.
Yeah.
Yeah, so we did collect gut microbiome data from the lesser longnose bat in Mexico. And what we found is that the gut microbiome is much more varied, much more diverse, and extremely different from the cave and bat skin microbiomes. We didn't see any impact of parasite occurrence on the bat gut microbiome.
And then, of course, the next thing I'm going to ask is, have you looked at the viruses to see if there's variation between the viral community that's being passed?
Stay tuned. We have some preliminary data, but not yet.
Thanks for the talk. That was excellent. I'm curious if, given the sophistication of the extended specimen repository idea, what sorts of things are you thinking about doing at University of Michigan to create more sophisticated scientists who are better data managers and better data users? Yeah, we're trying to build essentially a network of similar biorepositories currently across Latin America in collaboration with folks here and at the Museum of Southwestern Biology to do that exact training that you're talking about. There, not only do we need to think about our physical infrastructure and how we can completely store all these sample types, but also the human infrastructure. How are we training people to take these sample types and use these sample types? In that process, we also enable people across Latin America to have seats at the table, to be owners over their own country's pandemic prevention strategy through surveillance of wildlife for pathogens.
Any other questions?
Great talk. Yeah, in regards to the swabs and the the exponential increase of the holding these voucher types, do you need to keep all of them, I guess, after they're measured, or is there some type of is there a value in keeping some of the swab types for future use, dare I ask?
Yeah, so right now the challenge that we have in the microbiome field specifically is that our culturing methods are still getting caught up to our sequencing methods. So a lot of bacteria that we detect via sequencing, we are not culturable yet. So it is valuable to try and maintain swabs especially in ways that maintain live cells, so that eventually culturing could be possible, and then you can have a culture voucher of a bacterium. Until that day, it is important to keep those swabs, or at least a portion of them.
Any other questions? We've got a couple of minutes.
I was going to save this one, but since we have extra time. I'm curious about the third study. Since the bacterial microbiome is relatively in low diversity, were those metabarcoding abundances or were they qPCR abundances?
Yeah, great point. Yes, they were metabarcoding abundances, not absolute abundances. Yeah.
Do you think are the bacteria sort of suited for doing qPCR with them, or are they?
Yeah, there are absolutely assays where you can convert what you detect in terms of relative abundance from metabarcoding to absolute abundances using qPCR. And the value of that would be especially relevant for the situation where we saw differential relative abundances of Bartonella and Wolbachia in one species of fly compared to the other existing on the same host bat. So potentially relative abundance is not the correct measurement to use there, but by converting to absolute abundance through qPCR, we can say yes or no, this does have more of this pathogen, and then that would allow you to make conclusions about, okay, are there priority effects happening that dictate the abundance of certain pathogens? So are certain parasites more competent vectors?
Okay, I think we're at time. Thank you very much to both of our plenary speakers. We now have a quick 15-minute break, and then we're going to be going into our concurrent sessions. So we'll have 3 concurrent sessions, one that's running in here, and then the other 2 rooms that are set up at the back. So look at your program and see which concurrent session you want to go to, and we will reconvene at 10:30. Thanks.